Hello, everybody. Today we're going to be talking about Lab 11 and some of the theory behind it. Um, I have one that throws down. It is 10:22 p.m. Let's get at it. So Lab 11 is all about transformation. Specifically, what we're doing is we're taking E. coli K12, which is typically a pretty docile. It's in the name K12. Um, it's something that we use in different labs for just basic experiments. It's not gonna probably cause you any like actual disease, um, because it is just it's like a weakened strain. So, E. coli K12, however, does have the beta lactamase enzyme, and if you've been keeping up to date with lecture, beta lactamase is the enzyme that cleaves the beta lactam ring in penicillin, in ampicillin, in those types of drugs. And so it makes it antibiotic resistant to that class of drug. So that is the background on our microbe here that we are going to now perform a transformation on. So a little bit on what a transformation is. I know this is all things that you probably heard in lab, so I'm just trying to refresh. Um, Transformation is when we take DNA from a dead cell and put it into a live cell so that it can then express those genes. So we are doing bacterial transformation manually. We are making genetically modified organisms. Um, so one thing that Dr. B is going to stress a lot is that transformation can't happen unless the cell is competent. You'll hear that word a lot, and it is one of those key words that you have to keep in mind. Let's see. This is just a little tidbit. We do use recombinant DNA technology in order to modify other organisms. We do it with food, we do it with other bacteria. Sometimes when we're doing research with bacteria, we see uh, how how different strategies to make a cell competent work, if one's more effective over another, just things like that. There's a lot more that you can do with this rather than just, oh, wow, we did a lab, we made bacteria glow. We can do a lot more with recombinant DNA technology. There's a lot of research and there's a lot of fields that can be dived into. So this is personally my favorite lab, just because I thought it was really cool that we took something microscopic and we just mixed a bunch of like basically clear colorless liquids, and then all of a sudden we put it with the bacteria and then it glowed green under UV light. I thought it was really neat. Um so this part, in terms of the exam, I'd say like kind of be able to logic your way through it. There, if we're talking about GFP, green fluorescent protein, it's a protein found in jellyfish. They already glow. So we're going to extract that. And okay, well, we have, a little bit of gene. We need to make a ton. So we're going to amplify it. PCR is a really common lab technique in order to make a lot of genes, or a lot of proteins very quickly. Let's see. You guys will learn a little bit more about gel electrophoresis. I will actually be running an assay soon, so I'll let you know how that goes. Then once we separate that DNA that we really want, we're going to we're basically going to put it in something that the bacteria is going to be like, yes, I need this. It's kind of like when you give your dog, like, or like a cat, for anybody who has pets, It's like when you give them like a pill from the vet, you want to put that pill inside cheese or something or some kind of treat so that they're more willing to take it up and they're more willing to hold on to that. So think of like that little bit of DNA that we want to put in as like the pill, and then the cheese or whatever around it is gonna be the stuff that we add. Let's see. So this is describing what the plasmid actually looks like. And so I went ahead and I kind of just isolated the big key pictures here. Um, so like I said earlier, we have beta lactamase. I have that here at the bottom for you. You won't need to know the chemical structure, don't worry. But you see this little box right here? We have that's the beta lactam ring. It'll go ahead and cleave it. The beta lactamase inside of the bacteria will cleave the beta lactam ring of the antibiotic, rendering it useless. Knowing that like where things come from is the best for this one because it's really easy to switch. If you say that the beta lactam rings in the bacteria, that would be incorrect. The beta lactamase is the enzyme inside of the bacteria. Just keep that in mind. So this Pglo plasmid, it's fun and all to memorize it, but just understand what's happening. That's why I much prefer this kind of like circle. It's a little bit hard to see what's going on. So that's why I really like this one instead. So you have your Ara-C receptor. And so when Ara-C is going to come in contact with the arabinose sugar, it's going to change its shape and induce that operon. The operon is going to be in a stage where DNA polymerase RNA polymerase can then duplicate that gene. And so when we change the shape, we're creating that PBAD promoter. When we change that shape later down on the line, we have the GFP protein that can now be encoded for. So that RNA polymerase is going to move down from the PBAD promoter. It's going to duplicate the GFP and create mRNA that the cell will now express. Because before, they didn't have several copies of this gene because that gene hadn't been turned on until now. Only in the presence of it will that gene turn on and be actively made. So that should be good there. Trying to think of like a key point that students mix up a lot. I think it's just mainly like you don't add Ara-C, you add the arabinose sugar. Once you add that sugar, it changes that starting point to make that PBAD promoter, and then you get fluorescence. Just think back to kind of like if you can go from here and say, okay, now I can think about like a question being asked of me. So like if Dr. B were to ask you, we put a positively transformed microbe onto like our E. coli K12 onto LB amp era. Well, if it grew and glowed, then I can say that, you know, there is arabinose sugar on there. Okay, the arabinose sugar must have turned on this gene. Or if you want to think about the other way of like, okay, well, it could, if it's transformed, it has the arabinose, it has the Ara-C promoter and it has the GFP. So when that arabinose came in, it turned on the gene. If you want to think about it like that, that's fine. There's just multiple ways to get to the same point and include all the information. I would definitely recommend telling these processes as a story. So if you can sit somebody down and you're going to be like, okay, I'm going to tell you all about Lab 11. Perfect. Go ahead and narrate the whole thing to them. If you can do that and you feel like you have good detail on it, then I would say you're pretty set for the exam, at least for bacterial transformation. Okay, so that should be good there. I really like that visual because it kind of gets this like muddied point out of the way. It is important to know that you have the three different things on here. You have your Arac for the arabinose, you have your GFP for the fluorescence, and you have your blodgene. Let's see, is there anything else that I'm missing here? Competency, heat shock, cold shock. Understanding what these do, right? Think about your pores for the heat and cold shock. That's how I remembered it. Because you heat it, your pores are going to open up. So the cell membrane is going to open up. It's going to be more permeable. Cold shock closes the pores. It's going to resolidify. There we go. I think that's everything. At least that's everything that I want to pull from. That's everything that I want to do here. Oh, I misspoke earlier. That's my bad. So E. coli has the potential to be antibiotic resistant to ampicillin and penicillin. Typically, the E. coli K12 is not. Sometimes in the lab we will see regular E. coli be maybe a little bit more resistant to those antibiotics, but the blodgene here makes E. coli K12 resistant to anything with a beta-lactam ring. That is my bad. I digress. So the expected results here will show you that the E. coli K12 doesn't have the blodgene and it's not ampicillin resistant. So first up, we have our LB plate. This is just nutrient, right? This plate, if you plate the negative, the Pglo negative E. coli K12, basically this is going to be your control. This is going to show you that the bacterial sample that you were working with was viable, meaning that it was live, that you were growing things, that they were healthy, they were happy, there was nothing wrong with them. So that any further results that you get, they were because you changed a variable. So then you can compare that to your positive and negative on the LB amp. So the LB, remember, is just nutrient. The amp stands for the ampicillin. So if you take a negatively transformed bacteria, so this has not gotten the plasmid, because it does not have beta lactamase, because this is E. coli K12, because this is does not have the plasmid, and because it doesn't have the blodgene and it doesn't have beta lactamase, because of those three things, which are all interlinked, it will not grow. It won't grow, period, because it is sensitive to ampicillin. However, we have another plate, same thing, LB amp, but it's positively transformed. If it's positively transformed, it will grow because it has the beta lactamase. Now that's going to show that that part of the gene is turned on. That's going to be all the time. It doesn't need anything else to induce that part of the gene. Like how we saw with the Arac and the Pglo, the GFP. Those things needed to be induced. This is just always on. So this is just exemplifying that, okay, we don't have something that's transformed and we do, and there's a difference. So this is kind of like, okay, yes, we positively transformed the bacteria. Sweet. This also says that no bacteria that weren't touched that were on the control that bacteria wasn't already having the beta lactamase. So that bacteria was sensitive to ampicillin. That's what these two plates will tell us. And then looking at the last place, we have LB, AMP, and ERA. So looking at the LB AMP, right, we have nutrient and then ampicillin. So we are expecting growth at least, because that's what we had with the LB AMP and the positive Pglo bacteria previously. So now we add the ERA C, or the arabinose sugar. See, it's something that a lot of people mix up because they're very hand in hand. And I'm just trying to keep it straight for you guys. So the arabinose sugar is going to be in the plate. The ERA C is inside the bacteria. The ERA C is going to come in contact with that arabinose sugar. It's going to change its orientation on that gene. It's going to make the PBAD promoter. And then it's going to allow RNA polymerase to go ahead and make copies of that gene. Later on down the road, we have our GFP, our green fluorescent. So if we have effectively turned on this gene, we're going to expect it to glow. So that is the other half. I would say that the Pglo plasmid that we're looking at here, it's got two parts to it. The ERA C and GFP are interlinked. The BLA gene is always on. So there's those kind of two halves to it. But yes, I apologize for misspeaking about the beta lactamase and the ampicillin, but I would like to reiterate that E. coli K12, as we get it from our stock cultures, it is sensitive, meaning that ampicillin will kill it. If we put it on a plate with ampicillin, it's going to die. That's how we get our E. coli K12. Regular E. coli is a little bit of a different story, but E. coli K12, sensitive. Once we give it this gene, however, the Pglo plasmid, the BLA gene on the plasmid is always going to be made. Always, always, always. So that BLA gene is going to code for the beta lactamase enzyme. That beta lactamase enzyme, if it comes in contact with ampicillin, penicillin, anything with a beta lactam ring, automatically the E. coli is going to be able to going to be able to live. It's going to be able to tolerate the presence of that antibiotic. So not only have we made a genetically modified organism in this lab, we have also made a antibiotic resistant microbe in this lab. And yeah, I mean, lab 11, I found it really fun. Because it's the serological pipettes. I feel like I'm in NCIS. It's just overall, I really like it overall. So please feel free to ask me any more questions you might have. I know this kind of turned into like a 20-minute video. But yeah, I don't think that there's a whole lot more detail I could add on that. Without getting super duper granular. But yeah, any specific questions you might have, I could probably address those in the Discord. But other than that, I will stop my share. Thank you guys for tuning in. I'm sorry this is a little bit of a late upload, but hopefully you guys will find this useful going into exam 2. Just a little bit of a side note, going into exam 2 and this weekend, this is going to be the most amount of time you have in order to fully study. So I would recommend looking at the things that I posted. Dr. B really likes those. She is also looking into making something more interactive for a lab review. Then I would also look onto Canvas and look at the exam review material. That is stuff that Dr. B pulls from That is directly from material that she will be using. Do I have anything else? Maybe just quick announcement wise. After you finish exam 2, floor it through your unknown and your micro project. Start cranking out all of that work. The earlier you do it, the more you get out of the way now, the better. And right after an exam is perfect because that's going to be your lightest load per se. When you after an exam, you usually don't have anything to do for a little bit because you just barely have started covering new material. There's kind of like a pause in labs because you didn't have a lab that day. Um, so that's my recommendation. And if you do have questions regarding those projects, let me know. I can't review full unknowns or micro projects. But if you show me, I can give you a little bit of guidance, a little bit of advice. I can say, well, maybe on your micro project, if you're doing like a pamphlet, well, remember, you might need a little bit more space here. Your resources are too long here. I can kind of just give you some general advice, but I would recommend honestly just going to Dr. B during office hours when you have like the final draft and then making those small tweaks if you want to get that 100% A. But yeah, other than that, that's all I have for you guys. So good night and have a great weekend. Happy studying. And as always, I'll be available on Discord. All right. Bye, guys.